Good evening, everyone. It's a pleasure for me to introduce my friend and ex-coworker, Dr. Maria Bautista. Maria got her PhD from University of Illinois, Urbana-Champaign, studying viruses in the hot springs of Yellowstone. And after that, she moved up to Calgary to do postdoc at University of Calgary, studying hydrocarbon degradation in the Arctic Ocean. And subsequent to that, now Maria is a research associate at the University of Calgary, and she's part of a team studying wastewater-based surveillance for COVID and some of the viruses. So it's a delight to have Maria here and to hear about how wastewater can be an important scientific frontier to study human health. And I was wondering about one fun fact about Maria, and what occurred to me is Maria is originally from Colombia, and her dad is a coffee grower, and almost all of the coffee that Maria drinks comes from her dad's farm, where I guess kilos of coffee get put in suitcases and comes up with when either her parents visit Canada or she visits Colombia. So the coffee is fantastic. I could say that with the firsthand experience with it. So yeah, welcome, Maria, and we look forward to hearing from you about wastewater surveillance. Yeah.
Well, thank you so much for the introduction. Hello, everyone. I'm Maria. I do love coffee, and I also love viruses. So viruses are my favorite thing in the world. And so when they asked me if I want to talk about viruses for a little bit, of course, I said yes. But before I start, I want to just do a little disclaimer. So everything that I'll be presenting today are my own views and opinions, and they do not necessarily reflect the views or opinions of the University of Calgary and its employees. I just want to share a little bit of like the history and what has happened with this kind of field of science in the last 3 or so years, since the pandemic kind of happened and kind of changed the entire face of this field. And so I'll try to keep it brief. So at the end of the presentation, we can have some questions if you're curious about something in particular. If there's anything, I'm trying to not use a lot of like lingo or words, so it's accessible to everyone. But if you have any questions or something is unclear, please let me know. Okay, and so we went with this title because, when we go, to the bathroom, and we do either 1 or 2 or poop and pee or feces and urine. They have so many names. You can call them your favorite. We just flush the toilet and we don't think about it again, right? What happens to that waste? What is in that waste? As humans, we know that, urine is all of the waste, that just gets filtered by our kidneys, things that are toxins, things that are not needed. They just get removed from our body. And in poop, well, we have all the undigested food, a bunch of microbes, and who knows what else. And we flush the toilet and we forget about it. But then where does that go? And what can we actually get from that? And so if you think about a city or a town, every house is connected to the sewer system. And so when you flush your toilet, that goes into a pipe, that goes into a bigger pipe, that is connected to a bigger pipe. And at the end of the day, that all ends up in a wastewater treatment plant. So depending on the size of the city or the town, you might have more than one wastewater treatment plant. So I use Calgary as an example because that's where I live. In Calgary, we have 3 wastewater treatment plants that service Calgary and Airdrie and like Okotoks, like kind of the outskirts of Calgary. And we have one that is the biggest one that services like 800,000 people. So these wastewater treatment plants can take in the waste of a lot of people, but as well, not just what we flush, but what comes out of the dishwasher, what comes out of like industrial waste, lots and lots of carrots. I'm sorry, I know some of you are eating, but there are always carrots in the samples. So, at least people are eating their vegetables, which is good. And so how can we use this waste for something productive, right? When it goes to the wastewater treatment plant, it's going to get treated. But before it gets treated, what information can we get out of it? And so today I'm going to focus on how we can use this wastewater surveillance to detect viruses that are circulating within a community. Not to say that this can be used to do many other things. This is a very, very large and growing field, but I will focus on the virus aspect of it. Okay, so basically, what is wastewater surveillance? So we can monitor all sorts of things and within a population using this pooled wastewater. We can track infectious disease, the spread of resistance to antibiotics, pharmaceuticals, the emergence of new diseases, but also outbreaks of old diseases that we know and we think we have like in hand and we actually don't. And we can do it in real time and at a relatively low cost, which is also important. So you'll see why that is the case through the presentation. And so some of the advantages of this technology are that it is comprehensive, right? Like everybody that contributes to a sewer system is included in that sample. So it is inclusive. It is unbiased, and that is important. I'll put it in the context of COVID, right? Remember, not just 2 years ago when we were talking about symptomatic and asymptomatic people with COVID. So imagine if you are asymptomatic, you feel perfectly fine, yet you're infected and you can potentially infect other people. You wouldn't necessarily go get tested, right? Because you don't have any symptoms, but you're also sick. So those numbers usually get lost or don't get really measured because a person that is asymptomatic wouldn't necessarily go get tested. And so it is unbiased in that way that everybody, again, contributing to that sewer system is included in that sample. And it is anonymous. So as we were saying, a wastewater treatment plant can service thousands, tens of thousands, or hundreds of thousands of people. And so once you flush the toilet, that's it. Like that sample is there and it's anonymous. So, if for some reason you didn't want to get tested or know if you had like a positive or a negative, this is a way that, the result will be part of kind of like the pool that's out there. And everybody poops, right? Everybody poops, and everybody that poops flushes the toilet if you're part of a sewer system. And so that's why everybody's sample ends up being part of that collective. And so when we think about clinical samples, and again, the example here is going to be SARS-CoV-2 and COVID-19. If you remember early 2020, if you wanted to get tested, you had to, sign up, and then you have to drive through these places or line up for hours to get your test. And so one sample was one sick individual, right? And you could only get tested. At the beginning, everybody could get tested, but after regulations were changed and only people that had symptoms could get tested. And so that, I don't know if you remember how many tests were done every day. It was a lot of tests, and that was very costly, first of all, and also an extra strain on the healthcare systems because, they needed so many people, so many technicians, so many people like running tests all the time. Now, if you think about wastewater samples, one wastewater sample that gets analyzed basically just it's including so many people. It could include 800,000 people. It could include 1 million people. And so it is representative as well, as I was saying before, of people that have symptoms, that don't have symptoms, everybody contributing to that sewer system. But of course, this type of thinking, or this field, did not arise with the pandemic. It's been around for a long time. So people have been looking for viruses in sewage for a long time, right? When viruses were first described and people started to investigate them, they needed to understand how they were transmitted and things like that. And so, for example, during the 40s, and please keep in mind that the ethical regulations at the time were very different. These are experiments that today, of course, would not be something that could be done or approved. People were trying to investigate polio because polio was a big problem worldwide. And so they didn't really know if polio was an enteric infection, if it infected like the cells of the gut. And was it waterborne? I don't know if you remember, a lot of public pools were closed during polio outbreaks because there was a lot of kids that would get sick from water. So what was happening? Was it transmitted from direct or indirect contamination? So to pursue answers to these questions that were really pressing, and this was before molecular biology had really developed. So, the techniques were different. So in different cities of the States, in New York City and Chicago, and other cities, what they would do is they would take sewage, they would concentrate it, and then they would inject it onto monkeys to see if the monkeys were infected and developed polio. So this was kind of like the beginning of this field, kind of taking sewage and doing some trials on animals. Now, more recently, now in the 80s, we're talking about like late 80s, PCR is on the radar. So molecular biology is definitely starting to develop. People started using these technologies to try to monitor sewage. And so how did they do this? So originally, again, it was polio that was the target. And so the first sewage surveillance system was actually started in Israel. It was started in 1989 because they've had an outbreak. The last outbreak they had was in 1988. So they decided to start a program. So they would just monitor weekly or monthly the sewage, until nothing happened for like a long time, from 1989 to 2013. I'm terrible doing mental math, so I'm not going to do the math, but nothing, no polio. But in 2013, they detected polio, and it turns out there was a polio outbreak. They were able to trace it back to where it was in the community. It was a Bedouin community, and they were actually able to trace where that polio outbreak came from. So it had like come from Syria, it had spread to different countries, and it had started an outbreak. But thanks to this warning system, the Ministry of Health worked quickly. They vaccinated the public, and none of the infections resulted in paralysis. So it was really kind of worth having set up that system, even if you had run it for decades and nothing had popped up. Now, viruses that can multiply in the gut of humans or animals are known as enteric viruses, right? And so then they replicate in your gut. And these viruses are usually transmitted through the fecal oral route, which means they are shed in the stool of a host and enter the mouth of the next host by some means. I really like this definition because it says some means. So the means usually are poor hygiene. People don't wash their hands properly after they go to the bathroom, then they handle food, and then everybody gets sick. And I mean, this is something that we're all familiar with. I am in Calgary, so I think it was 2019. There was a big outbreak of norovirus from a shawarma restaurant. Unfortunately, I had takeout as the outbreak was happening, and I was one of the many, I think thousands of victims of that outbreak. And so, just having bad hand hygiene or something like that can infect a ton of people. And so these viruses have been monitored through wastewater surveillance for a long time. So norovirus, rotaviruses, polio. But so does this mean that only these types of viruses that are enteric and target the gut and are, transmitted via fecal oral are the only ones that can be studied through this technology? Well, you'll see for yourselves that might not be the case. I'm not gonna give the punchline away so early. And so the story begins, or maybe not begins, but gets interesting in the year 2020. Everybody thought 2020 was going to be a great year, and then everyone's life got turned upside down by a virus. Everyone's. So, in good ways and mostly bad ways, but we were not expecting a virus to wreak havoc like that, although scientists had predicted something like this for a long time. And so here we are, it's March 2020. The WHO has declared it a global pandemic, and researchers all over the world are trying to figure out, how do we detect this virus? How do we track it? How do we know it's here? Trying to find out as much as they can about this virus. And how to quantify it, how to detect it. So they looked back to some research from 2003 when SARS coronavirus 1 had appeared, and it was a small outbreak. It was not a pandemic. But they had detected or reported that this virus could be shed in the stool or the poop of infected individuals. And so, of course, people were wondering, is this virus also shed in stool? Of course, there were concerns if it was transmissible by stool, but also other researchers were like, well, if it's in stool, can we detect it in sewage? And so very, very early on in the pandemic, SARS-CoV-2 was reported to be excreted in the feces of infected individuals, not all of individuals, but many individuals. And again, just to say that this is not necessarily a virus that is transmitted like the fecal oral route. So it's a respiratory virus. So now after, 2-3 years, we know that in the feces, it's really not infectious. At this point, the virus has broken down, but we can still detect it. And because the virus, or a virus in general, is just a casing with information on how to make copies of itself and evade the immune system. So it's the casing, probably when it comes out of the feces, is a little bit broken. So it can't infect other cells, but we can detect the information, those nucleic acids, the RNA that is the genome of this virus. So we can detect it in the feces. And the first report of this virus, of detecting the signal, the genome of the virus in sewage, was from the Netherlands. And so this was a really important study because it defined, it was very simple, very small publication, but it was so good because it said a couple of different things. First of all, that actually sewage could be an early warning system because they had detected SARS-CoV-2 RNA in the sewage 6 days before the first cases were reported. And so this virus, when people get infected, they don't necessarily know they're sick. You can be pre-symptomatic. You can be shedding virus. You can be infectious. You can be making other people sick, and yet you can feel totally fine. And so a couple of days will pass, and then you'll feel sick. So this was detected in the wastewater, in the sewage. People could detect the signal before any clinical cases were actually reported. But the other thing that I think that is even more valuable or more interesting is that there was a correlation between the number of cases and the amount of signal detected in wastewater. So it wasn't just like a yes, no type of thing. Like, yes, we detect this virus. No, I don't detect this virus, the end, but more like the more of this virus that you detect correlates with the amount of cases. And, after so many years of pandemic, groups all over the world have shown this correlation to be really good and really strong. And so they have plots of the sewage, and where testing was happening really kind of religiously in different places, and people were actually doing clinical testing. They correlate beautifully. And so it is a way actually to track the levels of this virus in a population. And so, right, if we go back to how do we test for this virus, right? People would go get a nasal swab, sometimes a throat swab, that gets mixed with some reagents or chemicals that will break open the viruses if they're present and release the nucleic acids and remove all the gunk, the things that we don't need, and we have the nucleic acids. So again, this RNA genome of the virus or DNA of other viruses, right? In general, the nucleic acids, which encompass both DNA and RNA, are the fingerprint of each living or non-living, because viruses are not technically living, of any biological entity on the planet, right? They have nucleic acids as their blueprint. All of their information is there. And so scientists can design specific probes for each of them. So you can distinguish all of them from each other. You can distinguish variants. You can distinguish things, especially in this case, if you know what you're looking for. And so that's what PCR or quantitative PCR allows us to do. So we take all the sample, we take out all the nucleic acids in the sample, which includes nucleic acids from the humans and the plants and the bacteria that are in the poop. And just a total side note, but it is very interesting. The most abundant virus in wastewater is called pepper mild mottle virus. And it's a virus that infects peppers and tomatoes, some cucumbers. It infects a bunch of vegetables, but it is such a tough virus that, especially like the one in peppers, that it survives intact through the making of Tabasco sauce or any processed like pepper sauce. Then it's totally fine through the gut. And then people poop that virus. And it is so abundant in wastewater. So note aside, going back to our pathogen or what we are actually looking for, right? So then we know what we're looking for. We can put a probe that will only bind and produce fluorescence if our target is there. If our target is not there, it's not going to bind because it's very specific, right? And because it's also quantitative and it's linked to fluorescence, the more fluorescence you see, the more virus or of choice that you're assaying for is in the sample. And so that's usually, how the PCR work. When you go to get your nasal swab, and so you have a positive, it's like you have COVID, you have to stay home, you're gonna feel crummy, hopefully nothing else happens, but it was very stressful. And if you had like your negative, then you were happy to have evaded it for a bit longer. But this is how, we would find out. And so going back to the sewage, and kind of zooming in, right? If you are infected or someone in your building or in your neighborhood, like is infected, all of that is going to go down the drain. So here you see like little poops that have viruses, other don't. We also see some pee. Don't forget about the pee. I'm focusing a lot on the poop, but there are viruses that could be potentially shed in urine. And so Don't forget about them. I won't talk about much of these viruses, but they're still there. Just keep them in mind. This other complex, more poop is coming, and then all gets piled into this bigger pipe. And then again, it goes to the wastewater treatment plant. And so how these samples are actually processed is you take this raw wastewater, but you put a sampler that takes a 24-hour composite sample. So why is this important? Because as you can imagine, the flow of water on where people actually go to the bathroom changes. Some people are morning poopers. Some people poop at night. Some people poop at work. It's, it varies. And so if you only took one sample at like 10 a.m., let's say, then you would be missing all the other people that pooped at, 10:05, at 2 p.m., whatever. And so the way this is sampled is you place a sampler that takes a small volume every 15 minutes for a period of the 24 hours. And so here in this graph up top, you see the flow of water in a wastewater treatment plant. So you'll see that there are times of the day that there's higher flow because people are waking up, they're taking showers, they're going to the bathroom, they're getting ready for work, then it kind of lulls. And then at nighttime, again, a lot of like the flow increases because people get home, people do laundry, they're doing their dishes after dinner and stuff. So the sampler here will capture those 24 hours. And then that's the sample that gets analyzed. And so I'll use again, this cartoon I used at first. So again, we have one sample represents one individual that has symptoms. And then one wastewater sample represents a whole population. And the thing is that the sample gets processed the exact same way. So instead of starting with that swab, you start with the wastewater sample. And so you can see how this becomes a much more cost-effective. It's a complement to clinical testing. Of course, some people need to get tested and everything, but this represents 800,000 people instead of 800,000 people having to go get that test. And these parts of the procedure are pretty much the same, very similar, just, you have to kind of change bits of the procedure to clean up the sample a bit better because, of course, wastewater will have a lot more gunk than your nose. But from here on, the actual assays for qPCR and the probes that you use to go and probe out there and see if you find the virus are exactly the same. So for the cost of one PCR, you can assay a whole population. And so this became extremely kind of like it took off, like, I don't know what the word is, not popular, but it very, very powerful and very useful for people in different parts of the world to actually track how, like, what was the status of COVID in their kind of municipality and their population. So this is a screenshot of this website, which you should visit. It's really great. It's called COVID Poops. So it's basically a summary of the global SARS-CoV-2 wastewater monitoring efforts. And it was put together, this actual like dashboard by a university in the US. But the point here being this map is all the places that have like some sort of like wastewater surveillance system in place. So they're all over the world. I did this screenshot today, just make sure it was like really updated. So there are so many different dashboards and countries, different universities, and so many different sites that are being monitored for SARS-CoV-2. And so if you zoom in here, you will find that, of course, Canada is in that map. And so the PHAC, the Public Health Agency of Canada, has also put together kind of a dashboard with, and let me make this big, with wastewater surveillance. And so you can scroll, there are like different kind of summaries. And if you scroll all the way down, I'm sorry if I'm making you dizzy, close your eyes for a second. I'm just going to get down here. You have a list of all the different dashboards that are available in Canada. And so you can go by province and you can click on the different provinces and you can look at like each individual dashboard. So if you go, because we're in Alberta, the Center for Health Informatics, and so there is a tracker. And so the beauty of this is this data is for everyone. It's for public health officials, but it's also for the public. So this is reported real time. And anybody that wants to look at it and look at their state of their, their city, their town can do so and make informed decisions with this information. I use it all the time, right? Because I'm like, I don't want to get sick right now. What are the levels? Should I wear a mask, right? And sometimes I'm like, yeah, I'm going to wear a mask on the bus because I'm going to get COVID again. And I don't want to get COVID again. So this is here for everyone to just, look, and then you can click on the different places. So this tracker covers 84% of the population of Alberta. And you can look at the different places. And so this is pretty great. Now, and so as I was saying, right, like this field kind of started or, existed for a long time. And this is a screenshot again from today to get it as like fresh as possible of how many research publications have come out on this field, since kind of the first report that was like, late 70s, early 80s, not a lot of action for decades, but really in the last 4 years, it has really, really exploded. And in a way that it makes it, like we have learned so much about it. So that's been really good. Okay. And so again, we go back to the question we started with, right? Does this mean that we can only look for enteric viruses that are, transferred via fecal oral route? Absolutely not. As we saw with SARS-CoV-2, sure, this virus can infect gut cells. It can infect enteric cells. It's not very common. It is possible because of the ACE2 receptor, but research has kind of developed and grown with other types of viruses that don't necessarily infect the gut, and we can still detect them. And so I go back to the COVID tracker and I bring it. Again, this is the Alberta COVID tracker, but these efforts have been also kind of amplified in many provinces in Canada to kind of expand this to other respiratory illnesses that are, a burden to everyone right now. For example, we have flu. And so we have, I'm going to go back to Calgary. And so this is influenza A. So we know, because I think all of us here know someone that had the flu in the last month or so, right? Like people were very sick with flu. We had a big wave of flu. And this is, again, published in real time 3 times a week. So anybody can check, oh, how is the flu situation in my city? Should I, wear a mask? And this also helps public health officials to know if there are multiple things circulating at the same time, because the flu, COVID, and also RSV, respiratory syncytial virus, they are all respiratory illnesses, and they can present with like cold-like symptoms. And so you can know if multiple things are circulating at the same time, which was absolutely the case for at least Calgary. We saw a peak in influenza there was a peak also in RSV. So, people were sick with multiple things. So that also helps, again, like healthcare professionals to decide what they do, if they have to test, if they have to like identify what their patient has by knowing kind of the health of the entire population. And other things, of course, can be interrogated. For example, like the variants of SARS-CoV-2, right? Like variants are changing all the time. So that's something that you can also go to Wastewater and look and ask, what is the circulating variant in my, in my area? A lot of different groups are doing that for their specific places. And that is all I had for you. As I promised, I said it would be short and sweet, and I'm happy to take any questions and discuss any of the things that I showed you, clarify any concepts. Yeah.
Thanks, Maria. That was awesome. I wonder if we have questions among the audience. You could either raise your hand or you could type it in the chat and I can relay them to Maria. All right, we have a hand up from Carmen.
Hi, thanks for the talk. That was really cool. And I really like how you presented it. And easy to understand. Even my kids were excited. I have a question about how widespread this technology is, and if there's a sampling bias, like a rural urban sampling bias. Like I assume no one's capturing my COVID up here in Athabasca if I get it. Is that right?
Well, I guess I can't answer for every place in the world. I believe that for Alberta, a lot of places have been offered. And I think that if any municipality wants to be part of it, they can absolutely contact the program. It's a matter of logistics mostly, but there are many rural places that are being monitored. As long as there is a wastewater facility of some sort, a sampler can be installed and collected. I think it's more the matter of making that contact. Also can be difficult to make hey, do you know someone I can contact that person? And then that person can but I think that, I think that in Alberta, at least, it would be possible to even extend to more places that are willing and want to be part of it, for sure.
Thanks.
Awesome. We have comments and questions in the chat. There's a mention that I like that flu and RSV are being tracked and posted publicly too. So it's awesome. And we have a question. I believe it's a question. Yeah. Based on the Center for Health Informatics wastewater monitoring data, we have gone from long/wide COVID waves that extend over several weeks or months to very short waves that extend over only a week or two before there is a brief lull and then another spike in the graph. What does this suggest regarding the virus?
That's an excellent question, but I don't think I necessarily have an answer for you. I can, speculate, but I can't really tell you for sure what this would be, right? Like we have so many different variants, subvariants. The virus is changing a lot, and you see that again, this is all my speculation. Those smaller peaks, they don't seem to be as high. It could be because people have immunity from either vaccination, they just get mildly sick, they get over that variant, but we're gonna keep getting kind of reinfected over time, right? We know that vaccines protect us from severe illness, but not necessarily from getting sick. And so, but I think that vaccination also, that has been actually shown that the amount that that virus gets shed from people that are already protected either from previous infection or from vaccination is smaller. And so each person will contribute less to the sewer shed. a virus, so the peak overall looks smaller. So there are also a lot of people trying to do like modeling and like, statistics on how to link the amount of people corresponding to like the amount of like signal found in the wastewater. But that tends to be extremely tricky because, again, the virus keeps changing all the time.
Thanks. We have another question from Dan.
Hi there. Actually, I worked at the Bonnie Brook plant in Calgary for one summer, and I remember them talking. One thing that I remember is them talking about how people will put other things down the sewers besides, their poop and their pee, like oil. People dump illegal oil and chemicals and things like that. And like I was wondering, like with the RNA samples that you get, how do you know that like large numbers of RNA samples haven't come into contact with other materials that would like just destroy the RNA completely? And I'm like, I don't know anything about really the biological sciences. I don't really know about statistical analysis, so I don't know that I don't know at what level I'm asking for an answer here. I'm just wondering, like, how do you know that the sample is not somehow less, perhaps because there could be degradation of that RNA from chemicals, other things in the sewers?
Yeah, that's a great question, and absolutely is something that it's very important to take into account. So we, at least in Calgary, we have a method to measure inhibition. And so inhibition by whatever it might be, right? It could be like, as you said, chemicals, or it can be disinfectants. And so we add, once the sample is in the lab and it's already collected, we add a virus that we add ourselves that has a very similar structure to SARS-CoV-2, and it will behave very similar, and then proceed with the extraction and see. We also measure this virus we add. And so if the virus we add, we always add the same amount. If that amount is like off by something, then that sample gets investigated further. And I can tell you, for example, again, using the example of Calgary, we have found the effect of like samples with high amounts of like disinfectants. And so the signal was completely destroyed. And so, of course, those samples are not used for like reporting, because they are, of course, like not good samples. And every single sample is monitored for that. So every single sample that gets put on the tracker has been validated that it doesn't have significant amounts of inhibitors that would ruin the signal. But that is an excellent question.
Awesome. The next question we have is from Robert.
Hi, thanks very much for the talk. It was very well done. I have two questions. One is the China right now is supposedly underreporting COVID cases considerably. And from your map, it was a bit small, but I was wondering, is there one, say, on the Yangtze, which only comes from China, or is there one from Hong Kong that will give an indication of how COVID is doing in China?
That's a great question. I don't know, but because this website, so COVID Poops 19, you can just go to the tracker and it'll link you to the actual tracker. So if you're curious, you can just have a little research project of your own.
All right. The second one is a simple one about viruses. They're not alive, but we always talk about killing them. As a virologist, what do you say about incapacitating viruses other than killing them?
Yeah, that's more like a philosophical question.
It's always something that bothers me as a biologist.
I usually say inactivating them, right? That they're no longer able to infect new hosts. But yeah, killing them. But, if you look at like a package of Lysol, it says kills 99.9% of influenza A. So, and I see there's a question in the chat from Richard. So since COVID is a respiratory virus, how does it get into the feces? What is the route it follows? That is a very good question. And so for SARS-CoV-2 specifically, it can infect gut cells because there are ACE2 receptors in the gut. But flu and RSV, they don't really get amplified in that way, but they get still into the feces. And so if you think, and it's kind of gross, what happens when you're sick? You're like swallowing snot and like all the time. So you're kind of eating the virus. And so it makes it through and then makes it out. And so when you compare kind of the level of signal that you get, like absolute values of SARS-CoV-2 versus like flu or RSV, if you look at the axes of the graphs, the numbers for RSV and flu are smaller because probably, right, the total number of viruses that make it through are smaller because the signal is not getting amplified in the gut. But SARS-CoV-2 can get amplified in the gut.
Are there other questions? I have a question. So you had mentioned that you checked the tracker, and I was wondering, what are you looking for in the tracker to determine, oh, I'm going to wear a mask today versus not, because not all of us are data scientists or data interpreters. So I'm wondering.
That's a good question. I mean, I personally, I mean, the numbers don't really tell me anything, right? Because as I was saying, it's not like a specific correlation of a number with number of people infected. I usually look at the trends. Is it going up? Is it really high compared to other peaks? Like if you look in the tracker, the peak for Omicron is so big compared to the first waves of COVID. It was really bad, right? Like so
Would you
many
mind pulling it up? Sorry.
Of course. Let me see what I did with it. Let me go back. I'll just click on it again because I think I closed it. There. Here we go. So this one here, this super tall one is the first, like, I think Omicron peak. And so these were the peaks before, right? When we were on lockdowns and stuff, and they look very low compared to this one. So, right, like I usually look at a data point and then on the next one and see, are they trending up? If they're trending up, that's when I'm like, especially like in crowded places, I put my mask on. But yes, definitely not the numbers because we can't really translate those numbers to cases, but just the trends.
We have another comment saying, so informative, you made the information accessible. Thank you.
Thank you.
I mean, while I live in Calgary, I work for Athabasca, and I was also looking at the map and I noticed that Athabasca is missing, and I wondered about that as well. Yeah. As like further north of Edmonton. So yeah.
Well, you know who to contact. Do you have a contact in Athabasca?
Do we have any other questions?
I just wanted to say that one of my relatives was like a public health officer, like a doctor back in the 1950s during polio. And he set up the first like public health like lab where he lived. And he would have been like, I think he would have loved, been fascinated by all of this. So, yeah.
That's very cool.
We have a question. Could the tracker link be put in the chat? Yes, of course.
I'll put it now.
I also have it open. So.
Okay, are you gonna put it?
This is for Alberta. If you're looking for a different region, then there is a Public Health Agency of Canada has a listing. Yeah. but I presume most of us are for Alberta. There is another question in the chat. Based on the trends, what do you expect to happen with this virus? I'm going to assume we're talking about SARS-CoV-2 or COVID-19 virus, and not the RS. Yes. So yeah, what do you expect? What would you think would happen with it?
I don't know. I have to say that I very much respect the evolutionary biology of viruses. I would say expect the unexpected. Right, as a virus that is here to stay. I don't see it going away. It's going to be, but I can't also say, oh, and it's going to, kind of be mild. It could have a very, like a mutation that makes it escape current vaccine. So kind of, we would go into a cycle again. I was reading some paper actually of like the 10 most influential people in science by the Nature journal. And one of them was a Chinese researcher that actually has this model to kind of stay ahead. And his model can predict really well what's going to happen with the virus. So it would be probably a good idea to just keep an eye out for his predictions. But I don't know, like, I don't know. Viruses are just doing their thing.
Well, you did mention it's here to stay. So I guess kind of like flu, we'll just get vaccinated, routine vaccinations. Yeah.
So
Sorry.
flu is a bit different because flu is a segmented virus. So SARS-CoV-2 has like one genome, like one single piece is the genome. So of course it will mutate by just, mutation, errors in replication. But flu is a whole other beast that makes it a lot harder because influenza A has different segments in its genome. And so apart from being able to mutate, and like a lot of RNA viruses don't have proofreading. So basically they copy their genome and they don't have any way to do any spell checking like we do with our DNA genomes. We have proofreading polymerases. So if they make a mistake, they can like check the template and be like, oops, I put the wrong one, like erase and put the right one. But a lot of these RNA viruses don't have, so they actually mutate at a much higher rate. But then the flu also can have all these, like these segments. And so if it happens, and it happens often, is that one cell gets infected by two different influenza viruses that have different segments. Something called reassortment happens. And so bits from one virus get packaged and bits from the other virus get packaged, and they create a completely novel virus. And so and so because they are changing so much more, it becomes much more like hard to know how the next flu pandemic will look like. So
I guess so the next question is, how does this compare with H1N1 evolve? So I guess what exactly you were talking about? How does SARS-CoV-2 evolution compares to H1N1?
well, I imagine I read the question kind of similar to what a flu virus will evolve very different because that like H and N segments will get like reassorted. And so it can happen like all of a sudden, basically, when a susceptible host gets infected by different viruses.
I have one more question. Do you think this tool or technology can find use also in our farms? So can we test I guess there is no wastewater per se or treatment plans for farms, but can it be tested for bird flu or swine flu and things like that?
Stay tuned.
Okay.
I don't have any concrete answers right now, but maybe it is a good like if you think about it, it's a good concept. I'm not sure how like it can be really implemented and if it's gonna work the same way. So that's what I'm saying. Stay tuned.
Okay.
Maybe it will, maybe it won't.
Do we have more questions for Maria? Okay. All right.
There were a
guess.
couple comments of
Sure.
Carmen saying that she had to go, but super talk Maria, and thanks Srijak for hosting. And Teresa said thank you. Allison said very interesting, thank you. So I wanted to thank everybody for joining in tonight. And I also wanted to thank Maria for taking the time to do this for us for Science Outreach. It was great to have you, and
Thank
we
you for having me.
we really enjoyed having you speak for Science Outreach. So I really appreciate it. Thank you.
Thanks, Maria, and thanks everyone for attending.
Yeah, thank you for having me. Bye.
Bye. Thanks,
Bye.
everyone.